Alright, we're going to begin talking in Chapter 5 here today, and we're looking at biomolecules. Basically, we're talking about large biological molecules like carbohydrates, proteins, nucleic acids, lipids, and we're still dealing with the chemistry part of it. And the chemistry part is how we are assembling these very large molecules, and we're doing it by using building blocks. It's really important for your understanding of biology that you know what building blocks make up what biomolecules. In other words, what monomers are used to make what polymers. And the term monomer means that you have one thing. It could be a glucose molecule. It could be an amino acid in the case of building a protein. It could be a nucleotide in the case of building a DNA molecule. These smaller units are linked together through chemical bonds, and we'll be talking about those chemical bonds to build a larger polymer. And again, poly means many, so we have many mers, whatever a mer is, but I mean it's our building block, and so that's where we're going from here. When we talk chemistry for this particular chapter, we're talking about two fundamental types of chemical reactions that are involved. We have dehydration reactions, which are used to put monomers together and assemble them into a polymer. And then we have the opposite, where we're adding water in a hydrolysis reaction. Dehydration means we're taking water out. Hydrolysis means we're putting water in. And when we put water in, that is kind of the driving force to break that polymer down into smaller units. And these are constantly going back and forth in living things all the time, assembling polymers, taking them apart, and we watched that little animation which is basically talking about this process of dehydration and hydrolysis. So here's the key. We have a short polymer, or maybe it's another monomer, and we want to link to it another part of the chain. Could be another monomer, it could be another polymer. I mean, we don't have to just put monomers together. We could put any number of molecules together, but the key is on one end of the molecule we have a hydrogen that we're going to be linking to a hydroxide on the other end of our other molecule. And in doing so, this hydrogen, this hydroxide are going to come together and they're going to form our water molecule. Now keep in mind we have this carbon that still needs a bond here. All carbons need 4 bonds, and obviously we're not showing that in this diagram, but there's a carbon here and it needs a bond. There's a carbon here and it needs a bond. And so we're going to get them linked together and thus give us a longer molecule, our polymer. The opposite, hydrolysis, is doing the opposite of what we did just here. We're going to take a water molecule, we're going to split it in terms of attacking this bond, and in splitting the water molecule apart, we've attached the hydrogen to one side and the hydroxide goes to the other side, and now we have separated that molecule and we've released some energy, which is probably the driving force behind what we're trying to get accomplished anyway when we do reactions, chemical reactions that break molecules down. The impetus for doing it is to get energy. Macromolecules and their different groups are found in numerous quantities in every cell. I mean, we're talking thousands and thousands of these molecules, whether the proteins, carbohydrates, the DNA molecule. There is an immense variety of these types of molecules and their functions and what they're used for. Carbohydrates are commonly known as sugars. The more scientific name given to sugars is saccharides. So you'll see the term monosaccharide, disaccharide, polysaccharide, and basically that is just sugars and how many monomers are involved in the production of that carbohydrate. The simplest carbohydrate is a monosaccharide, so we've already mentioned that, and we can just keep building from there till we get to our polysaccharides. And of course, our building blocks are glucose molecules. You see, it is a ring structure. It's a 6-carbon chain, and that chain has been folded over so that it attaches to carbon 5. And we have talked previously in chapter 5 about functional groups, so you will notice we have created off of our ring, our newly formed ring, a functional group, and this is a carbonyl, and that carbonyl, and we also have a whole bunch of hydroxides. This is significant because these hydroxides are going to make this molecule polar, as is this carbonyl. And of course, the oxygens are kind of the driving force behind that. So we have a molecule that is very soluble in water. That is significant because it allows for it to be transported throughout the body very easily. Keep in mind, your bloodstream is largely water. So as you are digesting food and breaking that food down, you're pulling out a lot of sugars, a lot of monosaccharides, a lot of complex carbohydrates, and a lot of these are going to be transported very easily as they dissolve in the bloodstream, and then they can be pulled out by cells, at various locations. Here are some examples of different sugars, and in aqueous solutions, when you start putting these linear skeletons into an aqueous solution, you find that a lot of them fold over and they start to form rings. So monosaccharides then are building blocks for larger complex carbohydrates, and probably more importantly in terms of living things and what the function of a monosaccharide would be, here is our straight chain glucose molecule, C6H12O6, and it's demonstrating, and notice this is a reversible reaction, so we can go in both directions, the formation of the ring as well as undoing the ring and putting it back into the linear form. So the abbreviated ring structure looks like this, and again we have our carbonyl group and our hydroxide groups, and one of the things that we talk about maybe a little bit more later on when we get into complex carbohydrates is the fact that glucose has isomers, and those isomers are distinct. Disaccharides are when we take two monomers, two monosaccharides, link them together. The covalent bond that we're using for this linking is called a glycosidic linkage. We have a monosaccharide, glucose, that we want to link to another monosaccharide, glucose. And it doesn't have to be glucose, it could be, fructose, galactose, all kinds of different monomers. But the point is, again, this is going to be a dehydration reaction. We have a carbon here with a hydroxide attached to it. We have a carbon here with a hydroxide attached to it. And when we bring these two hydroxides together, one of the hydrogens is going to be stripped off, and it's going to combine with the hydroxide to give us our water molecule. Dehydration, we're taking water out. That then leaves this oxygen empty-handed. It needs two bonds to take care of the octet rule. It's got one here, so obviously it's going to want to link to this carbon here to give us our glycosidic linkage, our covalent bond that's going to tie these guys together. And of course, with hydrolysis, we're doing the opposite. If we want to break this molecule apart, or we want to break sucrose apart, we're doing the opposite. We're going to input water, and then the hydrogen is going to jump on to this oxygen, and the OH is going to jump onto the other carbon, and on we go. This allows us to build polysaccharides. These large sugars are again used as energy, a primary energy source, starch. And these large molecules made up of glucose monomers are basically the big storage molecule for plants, so that when they go through drought or whatever, they can still break those molecules down for energy. In animals, when we start making large polysaccharides, this is the biggie for us, glycogen. This large polysaccharide is stored oftentimes in muscle, and this is what your muscles need when you're exercising, working out, is a large amount of glycogen. Look at all these chemical bonds that we can just start attacking and destroying. This truly is an important polysaccharide in humans because we're using this for our everyday life, for the contraction of muscle cells. We store a lot of glycogen in liver for different reasons, and oftentimes you're going to find these molecules located very close to the mitochondria. And again, when we get into the chapter on the cell, the mitochondria is a very important organelle. It's the powerhouse of the cell. Okay, well, where's the fuel coming for that powerhouse? Well, it's next door. It's all these glycogen granules that are being stored around it for easy access, and away we go in the production of energy. Plants oftentimes will produce another polysaccharide, different from starch. This one is cellulose. This is a major component of cell walls. Like starch, cellulose is a polymer. Like starch, it is made up of different forms of monomers. In this case, we're talking about glucose, the alpha form and the beta form. And again, we got a little video clip that is laid out here in this chart. It's not animated, but I'll talk you through it. The alpha form and beta form, the big difference is how we orientate this hydroxide. So on carbon number 1, the hydroxide is pointed down, and over here with the beta form, it's pointed up. That simple rearrangement of the hydroxide changes that molecule in such a way that it goes from being useful for energy for the cell to being completely worthless, useless to the cell for energy. So when we take these monomers and we start linking them together, we produce a polymer that is very loaded up with energy. This is your spaghetti, your pasta, that type of food group that is going to give you a lot of energy for competition once you get it digested. On the other hand, if we use a bunch of beta forms and link them together, we produce something that we cannot digest. It just passes right through us, useless. And so again, understanding isomers is a very important part of the deal here in biochemistry, which is really what we're talking about here. And these slides are just showing you an up-close and personal view of our polymers, both the alpha form and the beta form. These cellulose molecules are often held together and grouped into, like we were talking about, microfibrils, which give us this very strong building material for plants. Cellulose, made up of beta-glucose molecules, they are straight chain. That allows us to kind of stack them together and wind them around one another, which is what we're getting with our microfibrils. Cellulose, on the other hand, is going to be helical, and we talked about this in class. That means that it's coiled. A lot more difficult to start packing those together, and so starch is not a good structural molecule. It's not strong, it's not durable, it's not going to work in that way. Cellulose, on the other hand, will, because of the shape of the molecule. And this is probably a much better picture, trying to take this little section of the cell wall, break it down, and when you get to the microscopic level, where we're talking about the monomers that are being put together to form this cellulose, you can see how all these chains are being linked together. Hydrogen bonds, making it very strong, coiled, really a well-designed molecule for the job that it has to do. There are microbes out there, there are bacteria out there that cows and other herbivores use to their advantage. This symbiotic relationship allows them to take grass and other plant material that would not normally be digestible and get it broken down